facilities. And the way we maintain the viability is by checking the viability of each accession here. So basically this is a seed testing laboratory. So I have mentioned that in the active collection we are checking the viability of the seeds every 5 years. So every 5 years we take samples from the active collection and sow them as a standard germination test. And the same with the long-term collection. But for the long-term collection, we are taking samples and checking the viability every 10 years. So since we were able to maintain the seeds for a longer time, so the checking viability is for a longer period also. So it's a regular checking. So that's how we maintain the viability inside the storage. So there is a threshold germination percentage. So 85%, the germination of the seeds should not fall below the 85% of the initial germination of the accessions, which is I think the threshold for initial germination before they are able to maintain this, I think above 90%. So yeah, basically once the seeds have fall below the 85% of germination percentage, that accession would be regenerated already. So only high viability materials are stored inside. So yeah, this is a germinator that we use. So basically we sow the seeds. These are for phase. I think this is phase collection. So much smaller number of seeds. I think 50 seeds. 2 replicates of 50 seeds being sowed for germination for about 7 days first count, and then count them again as a second count after 14 days. So for the active collection, we I think sampled 100 seeds, 2 replicates also. So since we have more seeds in there, and then also there's also more frequent PGTs inside, so we need to check the viability always for 5 and 10 years. So this condition, the seeds are sown inside a 30 degrees Celsius, constant 30 degrees Celsius temperature, 100% relative humidity. Yeah. So basically favorable conditions for germination. So since you have favorable conditions, you are sure that ungerminated seeds would maybe a sign of non-viability, so meaning dead seeds. But also to be sure, after 7 days, these seeds that do not germinate like this one are being dehulled as so. So they dehull it, remove this hull, and then sow them again for another maybe 7 days. Then see if they will germinate. Because this is just to check whether there is dormancy or not. So if it is dormant, so it would germinate. But if it did not, we're sure that the seeds are dead. And then the germination percentage is counted, and that would give us an idea of how the particular accession is doing inside the storage. So yeah, question so far. Also aside for this one, another important protocol is that every seeds that is taken out from the storage is being after ripened, dormancy breaking treatments after ripening in a 50 degrees Celsius temperature oven for 7 days. So that is just to make sure that any predominant dormancy is removed because due to long-term low temperature storage.
50 degrees, 7 days.
7 days, yeah. 50 degrees.
50.
50.
7 days.
Yeah. That's a dry heat treatment that we do. Dry. No, basically no. 50 degrees. That's just because as we can say, the seeds are have long been sleeping inside in long-term medication, which is called. Yeah, based on literatures that we follow, protocols that we follow, that is an optimum duration. Yeah. Really, once you go beyond 50 degrees, maybe you damage the seeds already.
So what is a normal problem if a wild type species, if you store it? A long-term storage, the problem of occurrence of regermination and problem.
Yeah, especially for the wild rice species, we have known that these wild rice species are very dormant. They have a deep dormancy, so that's being a problem also that we have. So that's why, aside from these day-to-day protocols that the gene bank do, GRC also have the seed physiology group, which conducts seed-related studies, which I happen to belong. So I'm a researcher of that group. So what we do basically, our main focus is on seed drying. So we're checking if that drying room is efficient. So we do seed drying experiments, different temperatures, different treatments, and also longevity studies, accelerated aging studies that we do on these different materials. So also just to check if these protocols that we currently do are optimum and maybe efficient. And also, especially the wild rice species, as you have mentioned, it's been a problem, the very deep dormancy that we have, because we could not handle or do much if these seeds do not germinate. So a lot of materials, especially in the wild rice species, are being discarded already because we are not able to manage them well because of this predominant dormancy that we have. So one of our experiments is geared towards dormancy breaking experiments, which I think this machine is running. And this is an ongoing experiment for the wild rice species. So we are playing with the after ripening duration. So as of now, currently the protocols that we use for the wild rice species, we just follow the protocols that they use for the cultivated rice. So we are not sure if that is good for these materials. Maybe not. So these are very diverse materials. So maybe they should have a specific treatment. So like this experiment is prolonging the after ripening period. So for the standard protocol is 7 days. So what we're doing here is
Are you maintaining any particular temperature or
yeah,
humidity?
this is a gradient plate actually. So you have a temperature gradient inside. So there is a different temperature conditions. Each petri dish is being subjected to a particular specific temperature. But also another main treatment is the after ripening duration. We increase it from 7 days, 14 days, 28, and 56 days after ripening. So I think I can show you some sample data here. Yeah, this came from previous runs. Yeah, this is from different species actually. So yeah, different species. So basically you see that each species have a different response to after ripening and also the germination temperatures. So you see here for these officinalis species, the 7 days after ripening treatment that the gene bank currently do does not really give much of the high germination percentage. But therefore, we also conduct viability tests to see if these seeds are dormant or dead. So we are able to prove that these seeds are still very dormant if you subject them to 7 days after ripening treatment. And then once you increase the duration of the after ripening treatment, maybe here 28 and 56 days, you see an increase in germination. So this color represents the germination percentage. The red color is 100%. Yeah, so maybe the ideal here is the orange and the red color.
So it really takes them 40 days and above sometimes.
Yeah, which is quite surprising. So, in general germination test, you consider seeds would be dormant after 14 days. But here we set the seeds here for 28 days, a month, and seeds are still germinating. And even after 28 days, some of them are still dormant, but still viable. So, and also, yeah, surprisingly, 56 days is quite a long period already for in 50 degrees Celsius, but it's quite effective. And also, if you notice that the seeds here are not dehulled, they are intact seeds, because one of the protocols that they do for the gene bank is they dehull these wild rice species every time they check the viability, which is quite very tedious. So you could imagine thousands of accessions being checked, so they dehull them. And this one, but still we do not have much germination. But this one, by just altering the germination temperatures and after ripening periods, so we have very high germination without even dehulling. So yeah, these are just some of the experiments that we do. So maybe to optimize the protocols that we currently have. Maybe in the future we could pull this information that we have and maybe create a protocol which is specific for the wild rice species, possibly for each species. So questions maybe so far? Yeah, so if not, maybe that's all for the gene bank. Yeah, so thank you for